Okay. So if you guys will look at the homework while I am. All right, it says if a woman who is a non-PTC taster, all right, PTC taster. So you're like, Miss Johnson, what the heck is a PTC taster? PTC is like a protein that some people can taste and other people can't. And so it used to be they put them on these little paper strips. And when we were in school back when I was a kid, they'd pass it out and they'd be like, hey, so if you taste it, then you have the dominant allele. If you can't taste it, then you have the recessive allele. Then you're homozygous recessive. So it was just a matter of whether or not you had that particular taste bud. And so that's kind of an old one. Nobody does that anymore because it's weird. Mainly because it's weird. So this woman is a non-PTC taster, so she's little t, little t. And she is heterozygous for hitchhiker's thumb. So that makes her big H, little h. And so then she has children with a man who's heterozygous PTC taster, big T, little t, but has straight thumbs, and so that would be little h, little h. So my first step here is that I've got to figure out what's in her egg cells, and so I would just put her phenotype here into a Punnett square. And it doesn't really matter what letter goes first, just be consistent. So I'll just put the H's first, whatever. And then I've got to put his little sperm cells into a Punnett square. Again, it doesn't matter which letters come first, but I'm going to go ahead and be consistent and put the H's first. Again, it really does not matter. Just be consistent all the way through. That way you don't get confused. And so there's my step one. And so here are her egg cells. Those are all the possibilities that are in her egg cells. And then here are all the possibilities that are in his sperm cells. I've just figured that out. And so there's step one. And so step two is the most tedious. Like I said, I like to just go ahead and just draw 4 Punnett squares next to each other. Seems the easiest route to take. Okay, and so I will put her little egg cells up here. Doesn't matter where she goes. So I'm going to put all her egg cell possibilities up here. And then I'm going to go ahead and put all his sperm cell possibilities over here. Okay. While I'm doing step 2, I can do step 3 at the same time. Okay, so I need to write down all the possible phenotypes. So I could have a PTC tester with hitchhiker's thumb. I could have a PTC tester with a straight thumb. I could have a non-PTC taster with a hitchhiker's thumb. Or I could have a non-PTC taster with a straight thumb. These are my possibilities. And so what I like to do is I like to go through and do the same thing. I like to do it at the same time. All right, so if I do the fertilization here, big H, big T, little t. Now I need to think about what that means. Big H is hitchhiker's thumb. Big T means PTC taster. Okay, so if the dominant is is there, if it is present, that means law dominant says that it's going to be expressed. So this child is going to be a hitchhiker thumb. In other words, hitchhiker thumb is when your thumb goes back like this. And be able to taste the PTC. So PTC, hitchhiker thumb, I've got 1. So I'm just going to put a hash mark there, or not a hash mark, but a tick mark. And then I'm going to go to the next one. And then, okay, so I have big H, big T, little t. I have the same thing. So that means that that person can not only have hitchhiker's thumb, but also taste the PTC. All right, so the next one, little h, big T, little t. Well, this is different. So this person does not have the dominant. That means that this person's going to have straight thumb, but they have the dominant for PTC tasting. So this is a PTC straight. So I'm going to put a little tick mark there. I'm going to continue. And that one is the same. Okay, so I'm just going to go ahead and put another. So this is a little different. I've got a hitchhiker's thumb here, big H, but this is a non-PTC taster. So non-PTC hitchhiker. So that's over here. Big H, little t, little t. Again, that is the same. Then I've got this one, little h, and little t, little t. Well, this person is a straight thumb individual and a non-PTC tasting individual. So that's over here, straight, non. And then little h, little t, little t. Again, that is the same. Going here, big H, big T, little t. Well, I know this person, hitchhiker's thumb, PTC taster, so that goes there. Big H, big T, little t. That is the same. Come here, little h, big T, little t. That's this one. Little h, big T, little t. That's this one. Big H, little t, little t. Okay, that's this one. Big H, little t, little t. Again, I've got hitchhiker thumb, but non-PTC taster. Little h, little t, little t. That's all recessive. Last one, little h, little t, little t. So I've got 4, 4, 4, 4. So that my phenotype ratio is 4 to 4 to 4 to 4. And that is number 1. Tedious, but doable. Are there any questions? Say that one more time. Yes, make sure you turn in the notes. Homework, you are completing number 2 and number 3. Do we understand? And I will make sure to post that video so that you can, if you need to go back and watch it again to go through and slow it down or whatever.